Okay. All right. So I just want to make sure I have the recording all set up. So welcome. We're doing this review for the first lab exam for Biology 150. My name is Dr. Etz. I'm one of the Biology 150 instructors at Montgomery College. And I'm just going to be going through this review packet. We'll go through it page by page. And as you have questions, please bring them up. If there's something that you really wanted to have explained or review in more detail, please don't hesitate to ask. You can either turn on your microphone and ask that way, or you can put it in the chat. I have the chat box right visible on the screen for myself. So if you put something in the chat, I will read it, and you can use that to communicate with me. All right. So let's start off with the first page, all about microscopy. And you should be familiar with all of the parts of the microscope, as well as the functions of each part. So there was a chart to fill out on the second page. So let's just start going through these letters. So what is letter A? What is this part called, and what is its function?
Letter A is an eyepiece, and it's designed for using of glasses.
Yes, very good. It's an eyepiece. It's also referred to as the ocular lens. But yeah, that's the part where you're actually like putting your face against it. And could you repeat what you said the function was? I didn't quite hear that.
It's designed for uses of crisis.
For uses of crisis?
Crisis, yeah.
Okay.
Glasses, yeah.
Oh, glasses? Is that what you said? Glasses?
Yes.
Okay, gotcha. Sorry, connection was cutting out a little bit. Yeah, so that's how you view your specimen. The purpose is to be able to look down, and the ocular lens, remember, also has a little bit of magnification to it. Does anyone remember what the magnification of the ocular lenses are? What magnification power is it? Yes, thank you. It is a 10x magnification power. And the other type of lenses are indicated here in B. So what do we call these ones?
Objective lenses.
Correct. Thank you very much. Yeah, that's the objective lens or objective lenses. And so we have three different, or some microscopes have three, some have four different magnifications there. So who would like to tell me what those magnifications are?
Uh, 4x, 10x, 40x, 100x.
Yes, very good. Yeah, so it's a 4, 10, 40, and 100. And so our total magnification power of any given microscope is going to be a multiplication of the objective lens power and the ocular lens power. So it's an important concept to keep in mind. And so these are just, various ways to just make your specimen look larger, right? This is how we magnify it and see greater detail of very small things. So these objective lenses are attached to part C. So what do we call that part?
Switch objectives.
So that the three objectives are attached to C, and part C allows you to switch between the objectives. But what is the name of this?
Nose piece?
Yes, it's called the nose piece, which is a little bit misleading. I tend to call it like a turnstile because, it kind of rotates around. So this is how we can switch between objective lenses. And All right, so if we're moving down, so we have, let's look at D, and on top of D is J. So J are like these little metal pieces along the side. Get a better view over here. And then D is this black square here. So we're looking for D and J. What are those two parts called?
D is stage.
Correct. Yeah, D is the stage. And then what about part J? J is this metal region here on the stage.
The stage clip.
Yeah, those are stage clips, or you call them slide clips. But basically those little metal things that, what is their purpose, if you can elaborate on that?
It holds the specimens in place.
Yeah, exactly. So the stage is kind of the platform for your slide, and then those clips kind of hold it down so it's not moving while you are looking at your specimen. And then next we are looking at, let's say E. Let's do E, F, and G. So E is this kind of vertical part that's coming down here. And then F and G are referring to the two knobs on the side of the unit.
E is stage control.
Right, E is the stage control, very good. And then F and G. These pictures aren't very high resolution, and these ones are kind of dark, so it's difficult to see, but F is the one that sticks out further, and G is the one that's like closer to the unit.
Is it the Iris diaphragm?
It's not an eyepiece.
The fine adjustment knob and the coarse adjustment knob.
Yes, that is correct. Yeah, these are the two adjustment knobs for moving, well, moving the stage up and down, basically. So F is the fine adjustment that makes smaller movements. And then we have the coarse adjustment. So all three of these parts, E, F, and G, they're all, the purpose is to move that stage so you can get a better look at your specimen. So the stage control moves your stage kind of, like left, right, or like backwards, forwards. Just going to abbreviate those words there. So the fine adjustment is for making very, very small movements, very small increments of movement. And then those larger movements can be made with the coarse adjustment. And those fine and coarse are both for moving the stage up and down. So we can move the stage up, down, or then, like left, right, backwards, forwards, in order to change kind of like our field of view of that specimen. All right, so moving over into this region, so we have H and I. So what are these two parts?
H is field view diaphragm.
Right, so H is the, so it's the diaphragm, which is also part of, or it's like right above the light source. And can you tell me the purpose of the diaphragm?
To see specimen clearly.
Right, so a little bit more specific than that. So, we have our light source, our lamp in the base there. And then right above it is, it's like an aperture, almost like a photo camera that you can widen or tighten. and it's going to change how much light is coming up under the stage. So if the diaphragm is completely open like this, then as much light as possible is going to shine through your specimen, but you can close the diaphragm and make it a little bit darker, which is sometimes needed to increase the contrast of your what you're seeing. So the light source is obviously, producing light for seeing the specimen, and then the diaphragm adjusts the how much light is coming through. And to answer your question, Kumari, yes, you have my permission to record. I'm also recording this session, and I'm going to convert it into like a YouTube video, which I will post, and then I'll send a link out to the 150 professors. So they will probably share that with you, on your course pages or something. So you can absolutely record this. I'm recording it too. And what was I just saying? Oh, right. So the diaphragm changes how much light shines to the stage. So almost like your own iris and pupil, right? So when your pupils are very large, you're getting more light to your retina in your eye. And then when your pupils are very, very small, it's only letting in a little bit of light. And then what about part I? Part I is, oh, okay. So I can, I see your message, Atia. I will see what I can do about those permissions.
Condenser adjective.
Yes, the I is the condenser. All right, so I'm looking for where I can give you permission to record, like how I can allow that, and I'm not exactly seeing it right now. And I don't want to take up too much time doing that. So let me just click around for like 30 more seconds. Unless someone knows how to do that and they can like give me instructions on that.
You can just like tap on her profile. If you see her profile, there should be like 3 dots, and then you can say allow share.
Allow share. Okay. Oh, I see. Allow participants to. Oh wait, no, that's not it. So under the. Oh, I see. Okay, under the not the participants list here, but okay. So Kumari and Atia, you would like to record. Let me find you here. All right, I think I did it. Faria and Atia. Okay, let me do this.
Is this going to be uploaded on YouTube as well?
Yes, I'm going to convert this into a YouTube video once we're done.
Okay, thanks.
Okay, so I see you all want to record, so I'm just going to go through and allow for everybody.
As long as it's on YouTube.
Okay. Yeah, so yeah, this is definitely like, I mean, that's the reason why we record them, so that we can make them available to all students in all sections, because, a lot of people aren't able to attend these review sessions because of conflicts with their schedule, which is totally understandable. And I know one thing that we usually have is a like a kind of digital sign-in sheet. So you can sign in with like your name and your Biology 150 professor's name as well. Because we usually keep track of that. So you can, because some teachers give extra credit for attending these sessions. I never got a link to that. So if you want to right now, just in the chat, just write, well, obviously your name comes up anyway, but write your name and your whoever teaches you Biology 150. And I will go back through the chat log later and just kind of record it up, and I will like send that to the Biology 150 professor, so that if your professor sees that you attended this session, you can get the credit that you deserve. It's not a guarantee because not all professors do that, but I know a lot of them do. They want to encourage people attending these. All right, excellent. So let's get back on track. I think I got everything set up here now. Yes, so we were left off at the condenser. So what does the condenser do? It's a little bit similar to the diaphragm. It has something to do with light, right? Which is, you can kind of guess by its position because it's right under the stage.
That also increases the amount of light that goes in.
Right, so the condenser is not about, so it's not exactly increasing the light, but what it does is it focuses the light. So you can either have the light be like very diffuse throughout the whole field of view, or you can kind of really hone it in to make it like almost like a really sharp light in one spot. So the diaphragm changes how much light gets to the sample, or the condenser can focus the light. Okay, excellent. And then finally, part K down here. Hopefully that's pretty straightforward.
That's an on and off switch.
Yeah, that's our power button or the on-off switch. And so that's just what is going to, turn the unit on or off. Excellent. Excellent. And thank you all for putting in that information in the chat about, who you have for 150. So I will make sure to put that into like a little spreadsheet and send it along. Okay, let's talk a little bit about the microscope. And well, obviously, microscope is very important for studying biological specimens because most biological specimens are really, really tiny. Now let's go to this question about what changes do you think would happen as you go from a lower magnification to a higher magnification? So let's say we're going from 4x magnification to 100x. So what's going to happen to the size of the image? Is it going to get bigger or smaller, or is it going to stay the same?
It's gonna get bigger, and the diaphragm's gonna be.
Right, so the size of the image is going to get larger or bigger. All right, what about the intensity of light? Is that light going to get brighter, or is it going to get dimmer?
Dimmer.
Absolutely. Yeah, the light is going to, intensity of light, we'll say it decreases, it gets dimmer. The field of view, is the field of view going to get larger or smaller or stay the same?
Smaller.
Molar.
Right, it's going to get smaller. Depth.
Isn't depth like the 3D dimension of like the specific specimens? So is
Right.
it gonna be decreasing?
Right. The depth is, yeah, kind of like that. Yeah, I guess it would be kind of the vertical dimension. If you're like looking at a sample from the top, it's going to be like how deep can you see into your sample if you're going to a higher magnification.
Oh, so is it gonna decrease because it's increasing the specimen only showing the higher resolution?
Yeah, it's going to decrease. Yeah, you're exactly right. So the depth, they're able to see, yeah, it decreases because you are kind of like, since you're zooming in, you're not going to be able to see like as much of that in that dimension, in that like plane of space, basically. And then the final one is resolution. What do we think happens to resolution? Right, it is going to increase. All right, excellent. Very, very good. This question, I mean, it's just an effort to kind of get you to think about the differences between looking at things from lower magnification and higher magnification. So let's try to name one good thing, one advantage and one disadvantage of looking at a specimen at 40x. And then we can do the same for 400x. So at 40x magnification, what is one good thing about looking at a specimen at low power?
So for that, you can see like a lot. So like field of view is increased for it.
Yeah. Yeah, you can see a lot of your specimen. You've got a larger field of view. Exactly. And then what about a con? What would be a negative of that?
So the con will be the resolution is decreased as the image get blurry. I mean the specimen get blurry.
Okay, so we'll say like the level of detail, or the resolution or level of detail is less. So you can see more stuff, maybe you can see more cells, but you can't see as much detail of individual cells. All right, and then for 400x, what is an advantage?
The advantage for 400x is the opposite. So you can see the higher resolutions and details. However, the con is that you cannot see a lot of specimens. So the field of view is decreased.
Right, yeah, so the pro, yeah, you can see much more detail because you're focused in on like just one very small area, but you're not going to be able to see as much because yes, your field of view has decreased. The FOV decreased, less of sample visible. Great. All right, so on your lab exam, there will definitely be some calculations concerning the field of view. And so you'll be given some information like this, and you'll be asked to calculate the field of view at some different magnification. So first of all, what formula are we going to use for this?
D1, yeah.
M1, D1
Yeah, so the M1,
equal,
D1.
equal M2, D2.
Exactly, yeah. So how would we set that up here?
It's, I guess, 46,000 micrometer equal to 400 times.
Yeah,
Yes.
just our unknown, exactly.
Yeah.
So perfect, yeah, so we have it set up that we have the magnification with its diameter of the field of view on one side of the equation, and then we have the other magnification, and we're going to solve for that magnification's field of view. Okay, so what is the solution here?
X equal 46,000 micrometer divided by 400.
Let's see, like 40 times 6,000. Let's see, 24,000 divided by 400 equals X. And so what does that come out to?
600 micrometers.
Exactly,
Oh.
yeah, 600 micrometers or microns. That is our answer there. And, if it's the type of question where it's not multiple choice and it's kind of a free response format, then you will be expected to kind of write out your calculations like that.
Okay.
All right, so someone got 200 as their solution. So
What 200? What do you do?
okay, so let's try to figure this out. So 40 times 6,000 should have gotten 240,000. Okay, yeah, so maybe it was just entering it into a calculator incorrectly. Or if you try to do like 40 times 6,000 divided by 400, if you try to do it like all at once, sometimes that can mess up the calculation.
So what is hypertonic? Hypertonic means higher solutions.
Hypertonic with an R, hypertonic means there's more stuff in that solution. So it's higher solute concentration.
Oh, thank you.
All right, let's do some metric conversions. So 3 millimeters equals how many micrometers?
3,000.
Yes,
3,000.
that is going to be 3,000. Very good. And then how many millimeters are in 500 micrometers?
0.5.
Excellent.
0.5.
Thank you very much. Yes, 0.5. And then how many micrometers are in 4,000 nanometers?
4.
It's going to be 4. And then 0.25 millimeters equals how many micrometers?
50.
It's 250.
Yes. Very good. Okay, so as long as you know kind of the hierarchy of these metric prefixes, it's pretty simple. It's just kind of moving a decimal place around. And there'll be a couple of questions like that on your exam. All right, so here we've got some kind of specimen. Oh, these are LODS cells at 1000x magnification. And it tells us the field of view is 5,000 micrometers at 40x. So it's asking us to determine the length and width of each cell. So I think the first thing we have to do is figure out what this field of view is at 1000x. It tells us what the field of view size is at 40x. So we're going to have to use this formula from up here again, the M1D1 equals M2D2. All right, so if we have at 40x, it is 5,000 micrometers. And then we're looking at something at 1000x. Now we're going to solve for x to figure out how wide across, how many micrometers across this field of view is.
$200.
Yes, I got that as well. So 200 micrometers. So this is 200 across, all the way across that circle. So now we have to use that information to kind of guess or like estimate the length and the width of one cell. So you could do this using either that counting method or that percentage method that you learned about. So what do we think? So let's do the length first, because I think that's probably the easier one. So looking at this, how many cells does it take us to go across the widest part of this field of view?
3.
So yeah, so obviously we see like a little bit of a cell, one whole cell, and then another partial cell. So I would say that this part of a cell and this part of a cell kind of complete each other. So I would say it's more like maybe 2.
Oh, so you don't count like the part of the cell, you just count like the whole?
Right, you want to kind of estimate, like if you took one whole cell and just kind of like line them up end to end, yeah, how many would go across?
Oh, okay.
So yeah, sometimes you have to kind of play around with like these little pieces at the end and imagine like, well, you know, this isn't exactly a whole cell, it's just missing a little bit, but we have a little extra bit over here, so it's almost like 2 whole cells. So if we have 200 micrometers divided by, it takes 2 cells to kind of reach across. So that would be, so the length of one cell is approximately 100, let's say. And then what about what about the width? So this dimension, this shorter dimension.
Like 5 cells fit widthwise in diameter, so 200 micrometers divided by 5 would be 40 micrometers.
Yeah, excellent. Yeah, so 200, and then if we're going, you know, let's go in this dimension or this way. So it's like 1, got 2, 3, 4, and 5. You know, this is cut off a little bit, but then we also have this extra bit up here. So yeah, I would say it's about 5. You know, this is about rounding and guessing. So it doesn't have to be exact, but it does have to make sense. So yeah, that's about 40 micrometers. So about 100 micrometers in length and 40 in width. And, you know, as long as you have your calculations, then you'll get full credit, even if your answer is like a bit off, you know. So anything that involves calculating, it's always really important to show how you got your final value. Don't just put your final number. Make sure you have it kind of written out, you know, what steps you went through to get that number. All right, so that does it for microscopy. Does anyone have any questions about the microscopy part? Otherwise, we'll start moving into the organic molecules.
I have a quick question.
Mhm.
So do you have to add like a percentage for it, like for the diameter? So for example, in this case, the length for the 100 is gonna be like 50% of a diameter. Do you have to add that on the test, or is this like enough?
So this is enough. So this is both of these answers we got using the counting method, but what you're referring to is that other method that you can use called the percentage. And the percentage method, you know, it's just a different way to get the same answer or similar answer. So percentage wise, yeah, if you're looking at the length, and you could say, well, like this one cell takes up about 50% of the field of view. So we could say it was 200 micrometers times 50%. If you made it a decimal, it's 0.5. So 200 micrometers times 0.5, and you still come up with the same answer that we got up here. So different way to get the same answer.
Also, either way is fine.
Mm-hmm. Yeah, either way is acceptable. You know, like sometimes it's easier to use one method over the other, and it's really just up to your preference.
Okay. Thank you.
Yep. All right, that was a great question. So now let's go on to these organic molecules. So hopefully you remember the 4 major classes of organic molecules.
The 4 major one was protein, lipid, carbohydrates, and nucleic acid or nucleic
Yeah,
acid.
nucleic acid. Yeah, exactly. Yep. So basically these 4 classes of molecules make up everything that's alive. Or, all these molecules fall into like one of these categories. And so you did those different assays to try to figure out what these solutions contained. And we'll start with the carbohydrates. So what is a monosaccharide?
Single sugar.
Yeah, it's a single sugar, which you could also call a monomer, right? So single sugar or a carbohydrate monomer. And then it asks us why all monosaccharides and most disaccharides are reducing sugars. So what is it chemically or structurally about these molecules that makes them reducing sugars?
So I'm not 100% sure, but I think because reducing sugar has a carbonyl, so it caused like them to go back and forth from linear function to the ring function,
Mm-hmm.
causing them to have like, that's why it's like, I guess, why almost carbonyl?
Yeah, that's basically like the key word there is that carbonyl group. So the monosaccharides, the reason that they're reducing sugars is because these molecules, I'll write that instead. These molecules contain a carbonyl group. And just as a reminder, that carbonyl group is a C double bond O. So it's the carbonyl group that is what gives these molecules the ability to act as reducing agents. And I'm realizing now that these answers are left over from the last time I did a review session. So we're looking at what do the terms reduction and oxidation mean? And one way that I teach my students of how to remember the difference between oxidation and reduction, it's this little mnemonic device called oil rig. So if you can remember oil rig, oil stands for oxidation is loss, and rig stands for reduction is gain. And so what is being gained or lost here are electrons. So when we're talking about reduction, it means something is gaining electrons. So if a molecule is being reduced, it means it has gained electrons. A reducing agent is the thing that is donating those electrons. And it's a similar kind of relationship here with oxidation and oxidizing agent. So an oxidation is a loss of electrons. And if you call something an oxidizing agent, that means it is the one that is receiving those electrons that were lost by another molecule.
So I'm a bit confused about this. So it's both opposite. So oxidation and oxidizing agent are 2 different things.
Yes, exactly.
Okay.
So this is where the way that things are expressed, the way that they're written is very, very important to pay attention to. So just like it says here, a reducing agent becomes oxidized. So a reducing agent becomes oxidized. It's losing electrons because it gave them up to whatever was being reduced. And an oxidizing agent becomes reduced. because it has received electrons. So I know it's very confusing at first. It's hard to keep straight. But just think of, if the word agent is involved, if it's in the phrase, then, the agent is kind of performing that action. So a reducing agent, it is like allowing this other molecule to be reduced. So it's allowing this other molecule to gain electrons. And it's because that reducing agent has given up those electrons. So this is the kind of thing I would recommend just kind of writing this in your notes, maybe coming back to it like later tonight or tomorrow, and like reading through it again. And hopefully like with that repetition, it'll slowly kind of like sink in and start to make sense. But yeah, if you can remember oil rig, so oxidation is a loss, reduction is a gain. And then whenever you see that word agent, reducing agent, oxidizing agent, that should be like your clue that like, okay, now I need to really think about what is that agent doing? So if we go to number 11, we had the Benedicts and the Barfoids reagents for these solutions. And they actually were made of the same ingredient because they say they both had that, very blue color, but they reacted differently to these substances. So why was their reaction capability different? What caused them to react differently with these carbohydrates?
Acid.
Okay, one of them is acidic. Which one?
Barfoids.
Yes, yep, that's right. So the Barfoids is slightly acidic. Actually, it's pretty acidic. So it has a pH of about 4.5. Whereas the Benedicts is, I don't think it's totally neutral, but it's more neutral than the Barfoids. So because the Barfoids is slightly acidic, it's only going to react with certain carbohydrates.
Monosaccharides.
All right, and number 12, this is another answer I accidentally left in. Really thought I cleaned this up a lot and deleted all these answers, but in any case, all right, so number 12, suppose you have performed the Benedict's assay with glucose. And then it asks which molecule was oxidized and which one was reduced. So you have to think about what was, which one lost the electron and which one gained the electron. And of course, I have the answer here. So the copper ion gained an electron. So because it gained, that means it was reduced. And the glucose was oxidized, so it lost or donated an electron. And the copper ion becoming reduced, that's what caused that color change, which is what indicated the presence of a carbohydrate. So the copper gained an electron. From where did the copper gain an electron? We know it came from glucose, but what specifically, what part of that glucose was able to donate an electron? Why was the glucose able to donate an electron to that copper?
The carbonyl group.
Exactly. Yeah, it's from that carbonyl group. Yeah, so that carbonyl group, because of its position in like the terminal part of the molecule, it's in this perfect place to kind of, yeah, just donate an electron if there happens to be kind of an accepting or receiving molecule nearby. So the glucose lost an electron, and that electron went to the copper ion, which is what caused that color change. And I'm going to just turn off my screen share for a moment and go through this document really quickly and delete any more answers that I might have left here from my last review session. So I will bring the page up back momentarily, just hold tight for a moment. Yes. All right, I think I got everything. So I'm going to bring back the screen. The only thing I couldn't get rid of, I'm just going to get a little creative and try to cover it up. Perfect. Okay. All right, I'm coming back to you. And to answer the question in the chat, yes, this will be posted on YouTube. Because it's going to be a very long recording, it's going to take a minute to get it into that format. I know the last time I did this kind of review session, it took a very long time to upload the file and for it to be, formatted. But as soon, literally as soon as it's uploaded and I have a link to share, I will send it out.
Will you also post the link at the end when this is over at 9?
I'm not able to do that because I don't get like the link for that YouTube video until like I make the YouTube video and until like it's
Alright.
actually formatted. So unfortunately I can't do that. But since I have all your names, you know, all the information in the chat, I could, yeah, I can try to like go through and just make it an email, right, that I just kind of send out to everyone with that link. So I can do that as well. And my channel, I don't even know what my channel name is. It's not searchable because I don't really have much on it except for these reviews. But don't worry, if you are here in this review session, I will make sure to get that link to you. That is going to be my homework tonight. All right, so moving on. So we're talking still about glucose and about how it has not only a ring form, but a chain or a linear form as well. And this statement is saying that in a solution, these two forms of glucose exist in equilibrium. And what is that? What exactly does that mean? So how can we have like two different types of this molecule existing in equilibrium? What do we mean by, you know, they're existing in equilibrium?
I think it means that they have the same number of atoms in their structure, and because the carbonyl group is no longer available when they are in the solution.
Right. So that's an important part, that when in solution, so when glucose forms that ring, it loses the carbonyl group. So glucose has the C double bond O only in its chain form, not the ring form. So in a solution, you're going to have equal numbers or roughly equal numbers of molecules that are in the chain form and the ring form. So some of your molecules, some glucose molecules are going to be rings, some of them are going to be just straight linear, and they just kind of sporadically shift from one form to another. So you're, you know, in any given solution of glucose, you're going to have roughly half chain glucose molecules and half ring glucose molecules. So when they're in a solution, they will just kind of spontaneously switch. All right, so question 14, we're talking about fructose and glucose. So they both tested positive as reducing sugars. So that means they're both able to donate an electron, right? When these two simple sugars kind of come together and form a disaccharide, sucrose, that disaccharide is not reducing. It does not have that same ability as these two monomers, you know, by themselves. So why is that?
Could you repeat the question again?
Sure. So we're talking about fructose and glucose. These are two monosaccharides, and they are both reducing sugars. So they both have that ability to donate an electron. And when these two come together, they form a disaccharide called sucrose, but sucrose is not a reducing sugar. So why is the disaccharide not reducing if the two monomers that make it are reducing?
Oh, that's a good question.
That's a good question.
I think when they're gonna form the link, the carbonyl group won't be like existing. They're gonna form make the link. So like the carbonyl group gonna not disappear, but it's gonna be in the link. So they won't be able to give something or be a reducing agent again. I think.
Yeah. Yeah, that's exactly right. So when sucrose is formed, and I see Luke and Camille also got that answer. So when sucrose is formed, that carbonyl group that used to be available to give up an electron, now it's part of that glycosidic bond. So the carbonyl groups, well, yeah, they don't disappear, but the carbonyl groups become part of the glycosidic bond, and that means that extra electron is no longer available to be donated. Yes.
I have a question.
Mm-hmm.
So it's like sucrose are in a ring form? Because
Sucrose is not in a ring form. I don't think it is. I'm trying to picture it now, or maybe it is. Yeah, it might be like a 2-ring structure actually.
because you say it's like you lose carbonyl, and usually you lose carbonyl when it's a ring form, right?
Mm-hmm. Yeah, when it's a ring form.
I think it's a ring and linear form, sucrose.
To donate an electron is lost. So the electron is no longer available just because, yeah, what used to be that kind of like free carbonyl group on the end is just, and now it is, you know, bonded to make this covalent bond with the other monosaccharide, and so it's just not available anymore.
Okay, I have a quick question for number 13. I mean, not number 13, number 12.
Okay.
Where did you get copper ion from? For number 12, it said copper ion is reduced. Like, where do you get that?
So the copper ion, that's like part of the solution. That's what makes that Benedict's and Barfoid solution blue. Where is it? Let me see. Let me bring up our exercise 2 lab manual.
So will they give us this information on the exam that stated like this solution has like copper ion or glucose, or do we have to figure that out?
No, it'll say like Benedict's or Barfoid solution. So as long as you know like what those solutions are, like what it's referring to, then you'll be fine. So if you go back to the exercise 2 like lab handout on page 3, at the bottom of page 3, it talks about the Benedict's assay. And it says the Benedict's reagent has copper hydroxide. When mixed with a solution containing a reducing sugar and heated, the copper ion is reduced from cupric to cuprous form. And then copper then bonds with oxygen to form cuprous oxide, which is red, and forms that kind of precipitate that you see as a positive reaction. So that's where the cupric or cuprous copper ion comes from. Is that, I guess, when it's in solution, the copper hydroxide, the copper kind of dissociates from the hydroxide. And so now it's just like this free floating copper ion. And that's why it's able to like gain electrons.
Oh, so you kind of need to know like the Benedict and Barrow solution and how they were made to
Yes.
like understand. Okay.
Yeah, exactly. And, you know, all that information is like written in the introduction to all the lab exercises. So that's actually a great way to review for this exam, is just going back and reading through the introduction pages. Because it's usually not very long, you know, it'll be like 1 or 2, maybe 3 pages with some figures. And that will really help you kind of understand the concepts behind each of these exercises. All right, so it asks you to identify the monosaccharides and reducing disaccharides. This is definitely worth knowing. So what were the different monosaccharides and reducing disaccharides? What were the names of these molecules?
Glucose,
Yeah, glucose was one.
Fructose,
maltose,
maltose,
what else are there?
One of them starts with an F.
Fructose.
And galactose.
Yes, galactose.
Galactose, yeah.
How
Very
about
good.
lactose?
I'm sorry,
Lactose.
was lactose on there? I thought it was just galactose. So these are just referring to the ones that you had in lab, not just like in general, but don't think lactose was one of them.
Okay.
But there were some disaccharides that were reducing sugars and some that did not reduce. So what's the difference, like chemically or structurally, between reducing disaccharides and the non-reducing disaccharides?
Reducing disaccharides has carbonyl in it that non-reducing does not have it.
Yeah, so reducing disaccharides had an accessible carbonyl group. So that carbonyl group wasn't, taken up in some kind of chemical bond. It was still available to donate an electron. Oops. And I'm just reminding you here what that carbonyl is. It's that C double bond O. Yeah, what are the reducing disaccharides? Who can answer that question? What are the reducing disaccharides? Oh, we did have lactose in the lab. Oh, someone said lactose, and I said, no, I don't think we had it. No, we had lactose. I apologize. I'm just looking through my lab handout now. Yes, Alexandra, you are correct. It is lactose and maltose. So those are two of the disaccharides that were reducing sugars. So these are our lactose, maltose, we'll do it like this. So these were our, these were our monosaccharides, glucose, fructose, galactose. And then our reducing disaccharides, we'll just say, just abbreviating that for sake of time. Reducing disaccharides were the lactose and the maltose. And then that one non-reducing disaccharide that we had in lab was the sucrose. Okay, so if we put a whole bunch of monosaccharides together and get a polysaccharide, what are three names of polysaccharides and what are their functions in cells?
glycogen, starch, and
Cellulose.
Yeah, cellulose. So glycogen and starch have similar functions.
and give energy.
Yeah, they are the storage form of glucose in animals, glycogen, and then starch is the storage form in plants. So it's storing energy in the form of these glucose monomers. And then what about the function of cellulose?
Is it like the structural?
Yeah, that's a structural polysaccharide. And it's primarily for making cell walls.
I think there was like two separate ones for plants. So cellulose for cells walls, and I forgot the other one, which was for plants in general.
The other polysaccharide is starch.
Oh, starch. Okay.
Yeah. So speaking of starch, if you took a starch molecule and you completely hydrolyzed it, and hydrolyze, this is the verb form of hydrolysis. So if you had starch and you conducted a whole bunch of hydrolysis on this molecule and broke all of those bonds, what would you produce? So what would you get if you just broke apart the starch molecule?
Glucose.
Yeah, just a whole bunch of glucose.
Why is that the case?
Because that's what starch is made of. So starch molecules, starch is a very large molecule and it's just made up of a bunch of glucose just stuck together end to end. And, you know, it's slightly branched. So it has a couple of branches off of it of like more glucose molecules. But if you were to just break up all of those bonds, all you would have left is just the glucose.
Okay.
And this is why like starch is considered such a filling like energy source. So if you ever eat like starchy vegetables or something, like this is what happens inside of your body, inside of your digestive system, is you have, you know, special enzymes and stuff which are going to break up that starch molecule and release that glucose, which then can get carried through your bloodstream to all of your tissues and your cells, and, you know, it keeps you full, and it also nourishes your cells. Okay, so here we had like drop down menus, and I wasn't able to like take off the answers. That's why I just kind of covered them up with these boxes. But it's kind of like a multi-choice selection if you're on the PDF version of this file. So what are the answers here? So if a substance gives you a negative Benedicts, a negative Barfoids, a positive iodine, and a negative biuret, what kind of molecule is it?
Starch.
Yes, ma'am. Yeah, that would be starch is the answer there. And the positive iodine is kind of the giveaway because that is the only assay that we did for starch, right? So if it's positive for iodine, that's all it can be. It wouldn't be glucose because glucose would not give that positive iodine reaction. And if it were glucose would also give you positive for Benedicts and Barfoids. So for number 20, so if we have positive Benedicts, positive Barfoids, negative iodine, and negative biuret.
Glucose.
Yes, this one would be in fact glucose. Right, because it is a reducing sugar and it is a monosaccharide. So it's going to give both of these positive results and then negative for the other tests. All right, what about positive Benedicts, negative Barfoids, negative iodine, and biuret assays?
Maltose.
Yeah, this is going to be our maltose. Right, so it is a reducing sugar. but it is not a monosaccharide. So that's why we have a negative Barfoids result. And then the next one, it says a substance that is positive in the BioRet assay, negative in all others. Let's see, why can't it be lactose? I guess it could be lactose. Yeah, since lactose is also a reducing sugar that is a monosaccharide, or excuse me, disaccharide. So here, at least on my like PDF fillable version of this file, it doesn't have lactose here as an option for like the drop-down menu. But yes, technically you could answer lactose here. That would be correct. And okay, so the next one, positive in BioRet and negative in all others. I'm going to plug in my charger for a moment. Okay, Eric, I see you say a bit confused how iodine and BioRet determine the solution. So iodine, if I recall correctly, the iodine reacts to the branching of the starch molecule. And then the BioRet, actually, I think it's one of the questions down here, what the BioRet actually is reacting with. So we'll get to that in a moment. But these solutions are, you know, chemically, they react with only certain unique chemical qualities of a given molecule. So that's how it will, you know, only change color if it's a protein and not something else.
A protein?
And yes, the answer number 22 is protein, or in the case of this drop-down menu version, the choice was egg albumin. But yeah, this would be a protein. And then what if all of the assays were negative? If we performed every single one of these assays in this lab and we got no positive result, what would that indicate? What would that suggest?
Sucrose.
Yeah, so it would probably be sucrose or some kind of non-reducing disaccharide, because we don't have a test to identify that specifically. So it's just kind of like process of elimination. If it's not reacting with anything, then it's either like just plain water or it's going to be a non-reducing disaccharide like sucrose. So the next few questions gives us just a little overview of some of these assays. So the Benedict's assay, what does it specifically test for?
Reducing sugar.
Right, this is our reducing sugar reagent test. Okay, so it's testing for the presence of reducing sugars. A positive reaction produces what kind of color?
Orange or red?
Yeah, so anywhere in like the orange, red, kind of brown color of families, or family of colors. Yeah, it's sort of like this rusty colored precipitate that we identify as a positive for Benedicts. And then the Barfoids, so this is the other carbohydrate assay, right? And this one distinguishes between two types of carbohydrates.
Monosaccharides and reducing disaccharides.
Exactly, yeah. So this is how we are able to tell what is a monosaccharide and what is a reducing disaccharide. Beautiful. All right, so number 25, talking about the protein solution. can be identified by performing which assay?
The BIOL assay.
Yeah, it's a biuret, burette. I've heard it pronounced a lot of different ways, so that's all correct to me. And this solution, what color does it turn if it's a positive result?
Violet.
Or like violet.
Yeah, violet or purple if you like. So that is the color change that we look for a positive result. And what is it that the solution is actually reacting to? It's the presence of what in the proteins?
Bonds.
Yeah, specifically the peptide bonds that link together those amino acids in proteins. That is what is being kind of identified by this reagent, is those peptide bonds. Excellent. So again, if you kind of want to refresh your memory on the concepts or like the specifics of kind of what these chemical reactions are all about, then I would recommend going back and reading the introduction, like those first couple pages of the lab handout for that biomolecules lab exercise, because that has all of the detail in there of, you know, what is reacting and why, and kind of lays it all out. So I think revisiting that is a good way to prepare. So we always like to confirm in our students that they are, you know, proficient in using the scientific method and identifying, you know, variables and controls and such. So you will very likely have a question similar to this one, where it gives you kind of like this fictitious scenario of an experiment. And then it asks you to, you know, create a hypothesis and identify the variables and maybe even interpret the results of a graph. All right, so we're looking at the scientists that work for a company and they want to try out these different sun lotions, sunscreens. So here's the kind of setup. They take the same amount of lotion and apply it to a person's arm and expose that arm to direct sunlight. The amount of redness on the skin is measured every 5 minutes for a total of 30 minutes. And these are the 3 types of lotion that they are putting. So a lotion with a new sunscreen chemical, a lotion with like an already available sunscreen chemical, and then a lotion that has all the same ingredients except for the active sunscreen chemical. So what question are we asking in this experiment above?
I will say sun.
Chemical useful.
So I heard which.
I will say, oh, she's still talking. Sorry, continue.
No, it's okay. Which chemical is more useful is the one answer I heard. And then what could you add to this?
I was thinking of is Sun X an effective sunscreen?
Okay, that's I like that better. It's a little bit more specific. Yeah, so is Sun X a more effective sunscreen? Excellent. We could elaborate a little more on these questions if we want to, say which chemical is more useful at preventing sunburn, something like that. Or is Sun X a more effective sunscreen? Both of these are totally fine. All right, and okay, so I see this one is asking for the hypothesis being tested. So remember that hypothesis is a statement, not a question. And it's a statement that tells you how the variables in your experiment are related. So keeping that in mind, what kind of hypothesis is being tested here?
So for this one, if sun X, so if sun X is used, it is more effective than sunscreen.
Is more effective than sun-kissed. So this is a really good start. Sun X is more effective. Now we have to add a little bit more detail in. How are we measuring the effectiveness?
By the redness of the skin.
Right. So we can say Sun X is more effective at preventing skin redness after sun exposure than sun-kissed or than other sunscreens. Okay, so you want it to be specific enough that the reader understands exactly like what you are testing, what these different variables are, and what the relationship is. So you think that this lotion is going to be better at producing a given outcome than this other lotion.
Can I say Sun X is a better sunscreen than Sun Kiss?
You could say that too. So me personally, for my students, I always get on their case about like, I want them to be very detailed in their hypotheses. So I would say like, well, better, that term, Sun X is a better sunscreen. I would want you to define what we mean by better. So that's where you want to kind of bring in some of this information of like, okay, well, what are we measuring here? Like, how are we going to determine which one is better? And it's because we're looking at this like redness of the skin. So Sun X is better, it's more effective. And what is it actually better at? What is it more effective at? And it's all about that, skin redness after sun exposure. And of course, in addition to being kind of specific as to how variables are related, you also want your hypothesis to be testable and falsifiable. And I would say that we did fill that criteria. So, comparing how red skin gets after using these two lotions, that's certainly testable. And if down the line, some better molecule, some better lotion ingredient comes out, then this hypothesis would be falsifiable as well. So by falsifiable, we mean like, could it be proven false or, inaccurate by another experiment in the future? So if, sunscreen research develops so much that Sun X is no longer the most effective at preventing skin redness, if we can imagine that type of scenario, then like that means our hypothesis is falsifiable. So I guess to better explain this concept of falsifiable, you would never make a hypothesis that said Sun X is the best sunscreen ever, or Sun X is the only way to prevent skin redness after sun exposure. A statement like that would not be a hypothesis because that's not falsifiable. It's too definitive, and it's not leaving room for future developments or for future researchers to come along and say, well, actually, we can amend this statement, we can amend this hypothesis. So that's what we mean by falsifiable. And then by testable, we're just talking about, you know, can an experiment be conducted, or can data be collected to test the statement? All right, so predictions are based on the hypothesis, and these are statements that are written in the if-then format.
So maybe you can say if people start using Sun X, then they will avoid the redness of the skin more effectively.
Okay, so if people use Sun X, they will then they will have reduced skin redness. Okay, I like that. That's a good start. We can add in compared to people who use sun-kissed or other products. And yeah, that would be perfectly acceptable. Does anyone have another one that they want to share? Ooh, so Anne, I like that one that you wrote. If the amount of redness is low, then Sun-X will be effective. I think you need to switch those phrases. So put the last one first. So if Sun-X is effective, then the amount of skin redness will be lower. Redness will be lower. And you know, you can also add this to the end, compared to other, or people who use other products, compared to people who use sun-kissed. Compared. All right, excellent. Okay, so what are the two variables here? What is our independent? What is our dependent?
The independent will be the new sunscreen.
Okay, so independent variable. Okay, the new sunscreen. Do we agree with that? What do we think about that? Right, so we can expand on that a little bit. So not just the new sunscreen, but actually all of those lotions are our independent variable. So we could say type of lotion used. Right, so that's what's being controlled by the experimenter, by the scientists. They're determining which lotions are being used in this experiment. And then what is the dependent variable? So what are we measuring here?
Redness.
Redness.
Exactly, yes, skin redness. And we can be really specific here if we want. We can say skin redness after sun exposure. Yeah, that's going to be our dependent, and the amount of skin redness is going to depend upon which lotion was used on the skin. Okay, so how are we going to determine experimental group, negative control, and then positive control? So it's like talking about these three groups, which are listed up here.
Group 1 is the experimental group because we are testing this group with the new sunscreen.
Perfect, yes. So group 1, because they are receiving the new product. the new sunscreen. All right, negative control.
The one who doesn't receive a sunscreen.
Right, the ones with just the regular lotion. So I believe that is group 3. The lotion has no active sunscreen chemical. And then positive control.
Group 2.
Right, so why is that our positive control?
Because they are getting the known sunscreen.
Yes, the known sunscreen, right? So they're getting something that is already known to work. We already know it's going to give us a certain result. Okay. Excellent, very good. Okay, so based on the results on this graph, is SunX more effective than Sunkist? So here we have our exposure to the sun in minutes, and then the amount of redness. It doesn't give us a unit for that, but I think it's kind of arbitrary. Yes. Scroll back up a little bit.
It is effective. SunX is effective. The levels of redness are lower than the other lotion.
Right, so it's showing much lower levels of redness after sun exposure compared to the other lotions. Yeah, exactly. All right. So this graph is missing a title. So let's come up with a good descriptive title for this graph.
Effectiveness of sun and in protecting from sunburn or sun exit.
Okay, so effectiveness of SunX in preventing sunburn. Yeah, that would be a good one. So I see, and you said effective versus ineffective sunscreen. So one way to come up with a really good title is you want to tell the reader, you know, the relationship between variables. So you can kind of look to what the axis labels are to kind of help you come up with that. So rather than saying like effective versus ineffective sunscreen, you could also say like comparing amount of redness in 3 sunscreen lotions or something. Or, you know, effectiveness of SunX in preventing sunburn, something like that. So you want to make sure you bring in like what was measured so that the reader already knows like what is being shown in the graph. So not just that we're looking at sunscreens, but, you know, how did we measure looking at sunscreens? Well, skin redness or sunburn. So yeah, a lot of possible correct answers for this question. All right. Okay, so let's move on to osmosis and diffusion. This tends to be a tricky one sometimes because it's really easy to get hypertonic and hypotonic confused, and also like trying to remember, you know, in which direction water is flowing. So let's make sure we can clear it all up. Okay, so we're looking at these beakers that contain these like osmotic bags of solutions, and the beakers and solutions, they have these different molarities that are listed here. So which beaker contains solution that is hypertonic compared to the bag?
Pica 1 and Pica 2 and Pica 5.
Correct. Yeah. So 1, 2, and 5. So I've got this little drop down menu here. So in all of those 3 beakers, 1, 2, and 5, the solution, the molarity of the solution is greater than the molarity of the bag, right? So 0.6 is larger than 0.2. That means that there's more stuff dissolved in this solution, making it hypertonic. Same thing over here, 1.0 is greater than 0.2, and 0.4 is greater than 0.2. So those are our hypertonic beaker solutions. Now, if we were, I'm sorry?
I'm sorry, I just have one question.
Mhm.
So is the water gonna move inside the bag or is outside the bag, the water movement?
In these ones, in 1, 2, and 5, so water is always going to move into a hypertonic solution because water wants to dilute anything that's hypertonic. So it's going to move out of the bag and into the beaker.
Also, it always goes from lower solution to a high solution?
Correct. Yeah.
Okay.
So let's draw some arrows. Oh, I don't want that. Come on. There we go. Oops. Okay. Right. So see if I can make this smaller. There we go. Okay, so we got the water is moving out of the bag into the beaker. In 1, 2, and in 5. Okay. In which of the bags would you expect to have the least change in mass? So which one is really going to have like no net movement? Like water's going to be moving across all the time. Right. It's going to be number 3 because they are isotonic. Very good. And now it's asking us to arrange the beakers in order of mass of the bags. Okay, yeah, so let's just arrange the beakers in the order of the mass lost.
That will be Pica 2, 1, 5, 3, and 4.
That is exactly the order that I got as well. Let's just put a little text box here. Yes, so 2 has the greatest difference of between these 2 solutions. So the water is going to leave this bag faster and rush into this beaker solution because there's a greater deficit here between these 2 molarities. And then we have beaker 1 and beaker 5. So all of our hypertonic solutions are going to, the bags are going to lose weight. They're losing water to the beaker. Then the isotonic is not really going to change much at all. It's basically going to stay the same. And then beaker number 4, this is the only situation where the solution in the bag is hypertonic. So water is going to enter the bag. Let's put a little arrow there too. Nope. Not what I wanted. Okay.
So water moves from low to high solute concentration?
Exactly, yes. All right, very good. Okay, so next up we have, I think, a little bit of calculating. So I'll try to keep it like this so you can get these answers down if you haven't already. So if we're moving on to number 41 on page 7. All right, a dialysis bag, just like one of these bags, is filled with a sucrose solution and placed in a beaker containing 30% sucrose solution. The bag's initial weight was 15 grams, and after 15 minutes, it weighed 17 grams. Calculate the percent change in weight of the dialysis bag. And then it gives us another time point. After 45 minutes, the same bag weighed 20 grams. What is the cumulative percent change in weight of the bag at that point? Okay, so we've got two different sets of data here. We've got some time and we've got mass in grams. Time is minutes, so that is those are our units. So we have an initial weight of 15 grams. So at time 0, our bag weighs 15 grams. Then at 15 minutes, it weighs 17 grams. And then at 45 minutes, it says it weighs 20 grams. All right, so using this information, how are we going to calculate percent change? What is our formula for percent change?
It's the mass at the time you want to use, and then you subtract it by the mass at 0 minutes, and then you divide that by the mass at 0 minutes again, and then you multiply by 100.
Yeah, that sounds excellent. So we have a mass at some time point minus the original mass. And then we're going to divide that by the original mass. And take all of that and multiply it by 100 to make it a percent. I don't like the way I annotated this. I'm just going to fix it for my own, just to my own liking. So we have, I'll say, so the mass at some time point minus the original mass. So we do that calculation first and then divide that by original mass times 100. Ooh, thank you, Zoliswa. I hope I pronounced that name correctly.
Welcome, Professor.
Right, so if we're trying to calculate percent change at 15 minutes, see, I'm gonna try to put this over here. So our percent change, we have 15. So, oh, sorry, the mass at our time point is 17. 17 minus 15 divided by 15 times 100. And that's going to be 13.3%. Oh, and I see you've already given us the next one too. Excellent. And that equals 33.3%. All right, so does everyone understand how to do that calculation, how we set it up, and then how we were able to get these numbers? If you don't understand, if you're still confused, please speak up now. All right, so I'm going to take that silence as a very good sign. Let's just keep on going. And so this is definitely a problem. This 41 is definitely a problem you'll see on your lab exam. So you'll want to memorize this formula if you haven't already. So just make a little mental note of that. Okay, so next we have some dilution calculations. So we are starting with a 20 molar glycerol stock solution, and we're trying to make these different dilutions that are listed here in this chart. And it's telling us that we need to make 50 milliliters of each of these dilutions. So what formula are we going to use for this?
C1 V1.
Yeah, exactly. It's going to be, yeah, our C1 V1 equals
V2.
excellent. All right, so what is our C1? Our starting concentration is this 20 molar solution, right?
Times V1. That's equal to 2.5 miles times 50 moles.
Right, so we're trying to figure out how much of this 20 molar solution we need. So V1 is going to be our unknown. So we have 20 times V1. C2, let's start with the 2.5 molar solution first. So 2.5 times, what's our final volume?
50.
Right, the 50 milliliters. And so now we can solve for V1. All right, so who has the solution to this one?
$6.25.
Excellent, yeah, 6.25 milliliters of that stock solution. And since we want our final volume to be 50, we're going to have to make up the rest of that volume with water. So basically we take our final volume minus what we added from the stock, and that will give us our amount of water to add.
For 43.75.
Yeah, 43.75 milliliters, exactly. All right, let's do one more of these examples. We can go through all the numbers if you want to, but we certainly don't have to. So let's do the 5 molar solution.
$12.5 now.
Yes, that is going to be 12.5. The final volume is still going to be 50.
amount of water will be 37.5 million.
Excellent. Yeah, 37.5 mils. Perfect. Oops. So again, the C1 V1 equals C2 V2, another really helpful formula that you should memorize for this exam. And once you know, once you understand where to put these values that are given to you, it's really easy to just get all these calculations done and fill out a chart like this.
Could you, I have a quick question.
Mm-hmm.
So for like the second one, could you like write it down in like the structure, like the equation? Because I'm still not sure about the second
Okay.
one.
Sure, yeah. So for the second one, I'm gonna just put up, let's get a text box here. Okay. All right, so our P1, oops. 2. All right, so our C1, our starting concentration is still that 20 molar solution. And V1 is our unknown. So we don't know how much of that 20 molar solution we need. But we want to make a 5 molar solution. And we know we want to make a 50 milliliter volume. So this is how we would set it up. So 5 times 50, and then we're going to divide it by 20. And that is what will give us our 12.5 milliliters.
Okay.
Yeah, so once you like compare it to this other formula, you see that like this is the only part that changed between the two formulas.
I see. Yeah, thank you so much.
You're welcome. All right, so we have even more calculations coming up. Let's, let me see. I think let's try to do this set of calculations and then we can take a little break, right? We've been sitting here for nearly 2 hours. So, all right, we're given this data table here. These potato plugs were weighed at 20-minute intervals. So it gives us these different intervals of time and how the weight has changed for these potato pieces, depending on what solution they were in. So now it wants us to calculate percent change in mass for each time point. So that percent change is the same formula that we used up here for this one, right? So percent change is the mass at some time point minus the original mass divided by original mass times 100. So let me make a copy of that box and I'll move it down here. Too fast. Okay, here we go. All right, so it wants us to fill out this whole table, so all the values. Yes, and we'll go back up a little bit. Okay, great. All right, so we know that if we try to calculate the percent change at 0 minutes, what is the percent change going to be?
0.
Yeah, it's going to be 0 because, you know, if you're comparing how much 5.3 is different from 5.3, it's not different at all. So there's no difference at the 0 time point because we're just starting the experiment. So the answer in all these columns, or in this first column, is 0%. So it hasn't changed at all because we're still at the first time point. And if you want to calculate it out, you 5.3 minus 5.3, well, that's 0. 0 divided by 5.3, still 0. 0 times 100, 0. So this first column is just 0% change. So let's do like at least the 20-minute column to make sure we know how to use this formula. Let's kind of put it over here. I'm going to change the size of this text. There we go. So at the 20-minute time point, so we're looking at 5.5 minus the original mass. So we end up with 0.2 divided by 5.3.
3.8.
Yes, it's going to be 3.8%. Okay, and let's do it for the next solution down here. So the 0.2 molar glucose. Well, we can tell just by looking at it that it's hasn't
0.
changed at all. Yeah. So that's going to be 0. For decimals, typically the rule is you can only round decimals like no further than what the data is, but then the decimals in the data itself. So since there's only one decimal point in the data that's given, you don't need to go beyond that when you're rounding. So you don't need to say like 3.776, whatever, it can be 3.8. Okay, let's do this next one. So this is, we're comparing 5 and 5.5. In the exam, if you are in a face-to-face class, then you will have to graph it. I don't know, because I'm not teaching face-to-face, I'm still teaching a remote section. So if it's a remote section and you're taking the exam online, there's no graphing. You will have to interpret a graph and know how to read it and use the data in the graph. But you only have to like actually make a graph during the exam if you are face-to-face. And so that will be done by hand. So you'll be given graph paper and there'll be rulers available, but you'll have to, kind of like make your own intervals and number it and everything. Okay, so what is the solution for this percent change? Looking at 20 minutes for the 0.4 molar glucose solution. Yes, you will be given calculators if you're in person. Yeah, they're like the really like basic little calculators, but those will be available for you as well. And there's scratch paper too, and I think there's like extra graph paper. So if you mess up, it's okay. Like you can start over again if you want. Great questions, by the way. It's always good to know what you're walking into, what you will have and won't have. Okay, so what is the solution for this calculation? Isabella, to answer your question, you could bring your own calculator if it is like the really basic, simple one. You're not allowed to use like a graphing calculator or anything, and you can't use your phone as a calculator. But yeah, if you have your own calculator, because I imagine they're going to sanitize these, in between sections taking the exam. But yeah, if you want to bring your own calculator and it's like the very like small basic one, then that is allowed. Okay, so this equation here, what is the answer here? What is the percent change for this one?
Negative 0.91%.
Correct. Well, it's actually going to be negative 9.1, I believe. So don't forget to multiply by 100 there to get it to a percent. But yes, the key here is that it's a negative number because the mass has decreased. So that's what we would put here. So negative 9.1%. Okay, great. So it seems like we're pretty clear on how to use that. If you'd like one more example, we can do this last one here. So the 0.6 molar glucose percent change. So here we are, this is our mass at time point is 5.0, mass, the original mass is 5.6. So very similar to what we just calculated. So we know it's going to be a negative percent. And thank you, Alexandra. Yes, that would be negative 10.7%. All right, so hopefully everyone is clear on that. So I'll leave the screen. Let's see, I'm just going to scroll down a moment. Okay, so I'm going to leave the screen up like this, and let's take a 10-minute break. So I'm going to pause the recording and turn my screen off and everything. And let's take a 10-minute break, just stretch, get some water. And so we will resume back here at, let's say 7:17. Right now my clock says 7:06, but let's just say 7:17, because by the time I finish talking, we might already be at 7:07. So 10 minutes, take a break, and I will see you back here. All right, folks, it was a very fast 10 minutes. I've resumed the recording, and it's okay if you aren't quite back yet. We're just going to keep moving along. Of course, you can always watch the recording later and kind of rewind if you miss something. All right, let me just delete this guy here. Okay. Okay, so we left off, we were calculating percent change in mass for this data table that it gave us. And now question 43 asks us, why are we calculating percent change in mass? Why can't we just compare the numbers as they are? Why do we need to transform them at all? So who has an answer for that?
We can know the potato cells. How much? I think we can know the potato cells gather water or lose water.
Okay, so we cannot know.
We can.
Oh, so we can know. Okay, gotcha. So we're calculating percent change in mass, so we can know which potato pieces gained water or lost water.
Yeah.
Okay. Well, I mean, the numbers tell us, like, you know, if we look at the mass data, that can also tell us which ones lost water or gained water. So what is the benefit then of calculating these percents here?
Hypotonic or isotonic.
So yeah, so knowing which ones gained or lost water can tell us the tonicity. So the real key here to like making these calculations, the reason why we do that is it's what we call normalizing the data. So because we have, like, you know, if you look at the 0 time point, they're all slightly different. You know, no matter how perfectly even you try to make these potato pieces, they're always going to be slightly different. And those little differences can, you know, can affect the outcome of the rest of the experiment and can affect these numbers. So in order to really capture what is happening in these potato pieces, and like you said, in order to really understand which pieces lost weight or gained weight in water, we want to calculate the percent change. So it's a way to, let's see. Why can't I get here? Okay. So calculating the percent change, we say normalizes the data because each piece has a different starting weight, slightly different starting weight, or mass rather. Yeah, so this calculation, it allows for more precise, analysis of these changes. So thank you.
You're welcome.
Okay, so we're not going to actually graph this here, but we are going to talk about, what the graph would look like. So first of all, given this information, this data that we have, what are the two variables? So what is the dependent and what is the independent?
Percent change in mass is dependent variable.
Yes, exactly. So percent change in mass, oops, misspelled that. Right, and then independent?
Time. Time.
Not exactly time, but what are we, obviously we're measuring over time, the change in mass, but what else, like what other parameter here are we manipulating?
Solution.
Yeah, the solution concentration. Exactly. So we're looking at change in mass, percent change in mass in these different solution concentrations. And yes, we are doing it over time. So we could say that time is another of these independent variables, but it's not as critical as the concentration. So our independent variable, it asks us, is it quantitative? Is this a number?
Yes, yes.
Yes, absolutely. Solution concentration is quantitative. You can count it. You can express it as a numeric value. Now this next one, is it continuous?
Yes.
Yes, it is continuous.
From 0 minutes to 20 minutes, 40 minutes.
Right. So even whether we're talking about time or solution concentration, they are both continuous variables because continuous refers to, are there like an infinite number of possible points in between the ones that we have chosen to measure or chosen to like look at the data? So there are, time points in between 0 minutes and 20 minutes. And there's also different concentrations in between 0 molar and 0.2 molar. So that's what makes those variables continuous. And because our independent variable is quantitative and continuous, that's going to inform which graph we're going to make. So which graph is best to make here?
Line graph.
Yes, exactly. The line graph. Oh, there we go. So we're going to make the line graph. And it's because of these two criteria of the independent variable. So it's best to make a line graph. The independent variable is quantitative and continuous. Okay. So you would use a bar graph if your independent variable was not quantitative or continuous. So instead of being quantitative, instead of being a number, if it was a category, so maybe like percent change in mass in like potato versus an apple versus like a banana or something, like, so that would be, those would be categories of things. Those wouldn't be like a numeric value. Okay, and so we're not, like I said, we're not going to make a graph here. I don't really need to go through that trouble, but if we were to make a graph, which one of these would be on the x-axis, which would be on the y-axis?
Y-axis and independent x-axis.
You were breaking up a little bit there. I didn't quite hear it, what your answer was.
I said dependent is the y-axis and independent the x-axis.
Yes, exactly right. So the dependent is always going to be on the y-axis. And the independent goes on the horizontal or x-axis. Very good. All right, so I'm going to scroll down a little bit more. And if you want to practice making the graph, then you can complete those calculations from that table, make the graph. So here it wants us to calculate the rate of percent change. So how quickly are each of these potato pieces gaining or losing water or mass? And the rate of percent change is the slope of each line of the graph that we just skipped making. So we don't need to go through the numbers. It's really just about knowing how to calculate the rate of percent change. So the rate of percent change would be the slope of the line. And if you are calculating it by hand, the slope is going to equal the change from the y variables divided by the change in the x variables. So basically, to calculate slope, you would need to pick two data points on one of the lines of your graph, and then use the XY coordinates of the data points to get your slope. Okay, so basically, you can either use the 0 time point as your x1, y1. So let's say x1, y1 could be your 0, 0 time point. So 0 minutes and 0% change. And then x2, y2 would be some other time point. So it could be, usually you want to pick like the furthest out time points. What is that, 40 minutes, I think? Let me see. Oh, sorry, 60 minutes. So 60 minutes, and let's calculate this really fast.
And 9.43.
Yeah, 8.6 for the 60-minute here. Oh, unless you're, yeah.
I got 9.43.
9.43. Okay, let's see here. Oops. Let's sort this out. Okay, so if we're looking at the 60-minute time point for the 0 molar glucose, so we're looking at 5.8 minus 5.3 divided by 5.3. That'd be 0.5. Oh man, did I enter it in the calculator wrong? It's possible. Oh, yeah, now I got 9.4. Okay, I don't know what I did before. I must have hit a wrong button somewhere. Okay, yes, so that is going to equal 9.4. All right, so let's use these two coordinates. So our x1, y1 is going to be 0, 0. Then our x2, y2 coordinate will be 69.4. So we're just going to scroll back down here. Okay, and now we can sort of place these values into this formula here. So basically, we're looking at 9.4 divided by 60. And yeah, I got 0.156. We'll say 0. Rather than 0.2, because that doesn't leave us a lot of numbers. Let's just say 0.16. 16. And yeah, so that's how you would calculate the rest of these slopes. And now if we were to complete this table and we wanted to graph this data, which one of these is the dependent variable? Which one is the independent variable?
So Professor,
Mm-hmm.
I have a question.
Sure.
Can you show us the 0 point more glucose, how much the rate of change? So how do you calculate that? Which 2 points you use? I want to know.
So you can use any 2 points. You can take any 2 points from the line of best fit that you,
right?
yeah. And so like typically you want to pick 2 points that are kind of far away from each other. So you get like a good spread of, if there was a line connecting these points, what is the slope of that line? And so I tend to use like the 0 minute time point and then whatever the longest time point is. It makes it easier to calculate using the 0 minute. Although I know some professors don't like that shortcut and they prefer to use 20 minute time point and the 60 minute time point to like compare to calculate the slope. But really, theoretically, you could use any 2 points as like your to fulfill that formula we were just looking at to calculate slope.
Okay.
All right, I want to highlight this. There we go. Okay. All right, so assuming that we went through these calculations and we wanted to graph this data, what would our different variables be?
Rate of change in mass.
Right, so that would be the dependent, because the rate of change in mass depends upon what?
Yeah.
Concentration.
Yes, very good. A course of correct answers: glucose concentration, or you could say solution concentration, doesn't matter. So our independent variable, glucose concentration, is it quantitative?
Yeah.
Yes, absolutely. And then is it continuous?
No.
It is continuous.
Mm.
It's continuous because there are an infinite number of possible concentrations between these 2 points that we have chosen. So we know that 0.1 molar glucose is between these two, but we could say, 0.15 molar glucose is between these two, 0.152 molar glucose. And like you could just keep, you could keep finding an infinite number of smaller and smaller incremental changes of molarity between these 2 points that we've chosen. So that's what makes it a continuous variable. Okay, so because our independent variable is quantitative and continuous, so once again, this would also be a line graph. So the reasoning behind that is the same that we stated before. Oops. Okay, and then of course, if we were going to make this graph, then the rate of change in mass, that would be our y-axis. Glucose would be on the x. And if you'll excuse me, I need to send a message very quickly to someone who keeps calling me. I need to tell them that I'm in a review session right now. Okay. All right. So this graph that you all had to make as part of your osmosis lab report, the graph that compares the rate of change in mass to the glucose concentration, what is special about that graph? What very specific information does it give you? I know it's hard to imagine, so let me fill in some information here. So this would be glucose concentration in moles. Okay, and then rate of change in mass right there. And this line, what it would look like, it would start up here. Oops. Let's just use this guy. Okay. This line would start up here and it would look like that. It would have this kind of negative correlation to it. So as the glucose concentration increases, the rate of change in mass decreases. It goes from being a positive value to a negative value. And then this point right here where this line crosses the x-axis, what is that point called? Exactly. Yeah, that is your isotonic point. Very good. Oops. Let's make this one a different color. Yeah, there we go. So this point right here, that's going to be your isotonic point. That's what makes this graph kind of necessary in that experiment, in that report. And what is the isotonic point? What does that mean? How do we explain what that is?
The solution are same in the cell and out of the cell.
Perfect. Yeah. So it is the concentration at which the solution is isotonic to the cell. Exactly. Okay, it looks like we are rolling into cell respiration next. So these videos are all very good, about how the lab is set up and how to analyze data for the lab, and then a little video about the difference between endotherms and ectotherms. So these are a great way to also review this, this particular exercise. First, let's kind of remind ourselves, what is this cell respiration, or here it's calling it aerobic respiration? Where is it happening? And about how many ATPs are we producing with this process?
Aerobic respiration is the breakdown of fuel molecules using oxygen to produce ATP.
Yeah, that was an excellent answer. So, sorry, yeah, cell respiration is the breakdown of organic fuel using oxygen to create ATP. So where is it taking place?
In the cytoplasm.
Not in the cytoplasm. There is a specific organelle where this is happening.
I think mitochondria.
Yep, there it is. Yeah, it's in the mitochondria. So we have a specialized compartment all for this cell respiration mechanism. So that is the mitochondria. And about how many ATP molecules are we going to achieve from one glucose molecule? Yeah, very good, Anne. So glycolysis, the very beginning part, does occur in the cytoplasm. And then once we have the pyruvate molecules, those pyruvates get shuttled into the mitochondria, and that's where the rest of the respiration occurs. So we can add that in there if we want. So it takes place in the mitochondria, except for glycolysis. Oops. Got cut off there. And the exact number of ATP molecules from like one molecule of glucose isn't fully agreed upon among scientists. So you'll see values, it's usually around 30 ATP. Some people say it's more, some people say it's less, but you can be pretty safe by saying 30. Okay, and next it's asking us about fermentation. So what is fermentation? Where does it take place? Or maybe what organisms do fermentation? And how does it compare to cell respiration?
It's anaerobic respiration without oxygen.
Right, so the big difference here is that is anaerobic. And I mean, this could be seen as an advantage that, okay, you don't need oxygen around for this process to occur. But if you compare it to cell respiration, how many ATPs do you get from fermentation? Right, yeah, so that's the very good, Alexandra. So only 2 ATP are being made, and it's because the only processes that are happening is glycolysis and then something to regenerate NAD plus. Add that here. So it's anaerobic, consists only of glycolysis. And yeah, because it's only consisting of glycolysis, it can occur in the cytoplasm of animal cells, and it can also happen in bacteria, bacterial cells, yeast cells, all kinds of microorganisms. So a lot of different life forms are going to be using fermentation. So let's go over that summary equation for cell respiration. What are our inputs and outputs here? Don't worry about like the values for everything, but just give me the, molecular, the molecular summary. So what's the chemical formula for glucose? We know we need glucose for this, right? Perfect, thank you very much. So C6H12O6, that's our glucose molecule. And then of course, we're gonna need some oxygen. So if you want it balanced, you'll say 6 oxygen molecules per glucose molecule. So those are our inputs. And then what are the outputs here? Excellent. So we've got 6 carbon dioxide, a couple of water molecules, and some ATP. I'm going to amend this up here. I'm going to say 30-ish, just, you know, kind of 30. All right, perfect. So we are breaking down the glucose using oxygen, and we're going to come up with carbon dioxide, which is a waste product we exhale, a couple of water molecules, and then, of course, that energy that we've extracted from the glucose. What did we measure in the cell respiration experiment?
See the volume of oxygen consumed over time.
Right. So the volume of oxygen consumed over time. So how much these little peas and crickets were breathing. Very good. And here's the basic setup of that respirometer. I don't think we need to go through and label it because we're going to kind of talk about it down here. So why was this respirometer submerged underwater in that bath?
To seal the chamber and trap the air.
Right, so the respirometer was submerged to, yes, kind of seal the chamber and trap air into this pipette. And this was kind of the way to control how much air was going into this little closed system here. And then why was water moving in as time went on?
So oxygen consumed.
Right, so it was moving in because oxygen was being consumed by whatever was inside the respirometer. And basically as the gas was being used up, this pressure inside here was decreasing enough so that the pressure of the water outside could kind of push into the pipette. Let's say the pressure of the water outside exceeded air pressure inside. Okay, so the water is able to push in as that oxygen was being consumed by the little cricket. And this also brings into, well, you know, if oxygen was being consumed, presumably this cricket was also producing carbon dioxide. So what happened with that? Why wasn't that pressure keeping the water out? So carbon dioxide was eliminated from the experiment. How? How did we deal with carbon dioxide being produced inside of the chamber? Right, it had to do with this KOH, this potassium hydroxide. So the potassium hydroxide, the KOH, reacted with the carbon dioxide and basically turned it into a solid. So it was no longer a gas that was creating pressure, pushing the water out. So it was, yeah, it was turned into a solid by that KOH. And that's, I think, what is supposed to be represented here by this like yellowish mass. So potassium hydroxide was kind of like dropped onto that cotton ball. And then we had like fresh cotton above it to protect the organisms from the KOH, because it's not, yeah, it's not something you want to be touching. So as this cricket breathed out CO2, that CO2 was absorbed and turned into a solid by that KOH. And again, the details of that, like chemically what's going on and that reaction, you can read about that in the introduction to that lab exam, the lab handout. So again, you can always go back to those lab exercises and read the first couple pages of each of them, and it will help you kind of understand what was done and why it was done. So remember, there are two types of peas that were being used, right? The dry peas and then the germinating peas. So what was really the difference between them? What does that germination even mean?
Germinating is a pea that has a growing seedling which is plant embryo. These peas are doing respiration, containing starch.
Right, exactly. Yeah, so the germinating peas are actually like activated to grow. And so they are actively performing cell respiration. They don't look like they're doing much, but there's a lot going on inside of that pea once you soak it in water and give it some time to start that process. So they are performing cell respiration using starch that was stored there by its parent plant. And that's what's going to allow it to grow roots and stems and leaves and grow into a full plant. And that is also how seeds stay alive without leaves and chloroplast, is that they're kind of in like a dormant state. And then once you give them the right conditions with moisture and warmth, then they will start to grow. And they do that using cell respiration. Okay, so next we are calculating the metabolic rate of 10 crickets that weigh 2.5 grams and consume 2 mils of oxygen in 6 minutes. So we're given some information here, and now we have to use it to calculate something. Now we need to remember what the formula is for calculating metabolic rate. And I think it's easiest to, before you think of the formula, just think of the units. What are the units for metabolic rate?
Hour per gram.
Yes, the first part cut out, but I think I heard you say mils per hour per gram. Yes, because it's a rate. So we're looking at something over time, but metabolic rate also takes into account the mass of an organism. So we're looking at milliliters of oxygen consumed per hour per gram of weight. Excellent. So if you can remember what the units for metabolic rate are, it kind of leads you into figuring out this equation or this formula. Okay, so first we have to take the 6 minutes and convert it into hours, right? So we know these crickets consumed 2 mils. Two mils in 6 minutes. And I see So Liswa has already calculated that out for us. So we're looking at 0.33 mils per minute and multiply that by 60 minutes per hour. So that way we can get this into mils per hour instead of mils per minute. So we're looking at 19.8 mils per hour. And now we have to just divide this by our mass, which is 2.5. Let's see. Right, divide that by 2.5 grams. And yes, that's going to give us 7.92. It's fine if you round it differently and you got 8, like a round number like 8 mils. That's all right too. So 7.92 mils per hour per gram of weight. All right, so does everyone understand how we did that? Or is anyone confused and needs a little more explanation? No, in this case, the number of crickets does not matter. And that's what bothers me about this question actually, is that, yeah, it tells you that the number of crickets, which is actually irrelevant in this case. So we don't care how many of them are in there. We just care how much they weigh. So I don't know, maybe that's put in like purposely as a distractor, but no, the number of crickets does not matter. We're really just concerned with per gram of weight. So whether it was 100 crickets in there or 10 crickets, it should theoretically work out to the same metabolic rate because it's per gram of, you know, living stuff, right? Yeah, so don't let the number of crickets throw you off there. All right, I'm going to scroll down a little bit. I'll keep this visible so we can still see it. But now it's asking us for the relationship between body size and metabolic rate.
The body size is bigger and the metabolic rate is higher.
Okay, so larger body size means higher metabolic rate. Do we agree with that? So if we look at our example up here, so if our body size or our body mass here is 2.5 grams, we get 8. But what if our body mass was 250 grams? What would happen to the metabolic rate then? If it was 19, I'm sorry, I didn't hear what, is that Hannah? Oops. So let's say it was 19.8 mils per hour divided by 250 grams. So what would that mean for our metabolic rate?
It's lower.
It would be lower, yeah. So it would probably move the decimal place the other way. So it'd be like 0.00 or 0.0792 mils per hour per gram. Right, so a larger body size would actually mean a lower metabolic rate.
Professor,
Mhm.
for my because I think for sometimes if the body cells is larger and they need more energy, so maybe the metabolic rate is higher. I'm not sure why it's lower rate.
Right. So, yeah, I can understand that if you're just thinking about it kind of logically, it makes sense that if, like a larger organism would, need more energy and would be metabolizing at a higher rate. The reason for this inverse relationship, trying to think of the best way to explain it. And I can't come up with the best way to explain it without using like an anecdote of like, like small mammals compared to us, that small mammals tend to have a much higher metabolic rate. They go through energy much faster than us like humans, or, our metabolic rate is faster than say like an elephant, which is like much larger in mass. And I can't figure out how to explain it, but I just know that it's the inverse relationship, that they are not as efficient as using energy, I guess, the smaller mammals or smaller animals. And so that's why they tend to have higher metabolic rates. I wish I had a better way to explain that, but I'll be honest, I've had a very long, busy weekend and my brain is kind of reaching its limit. So I would say maybe reach out to your own professor, your Biology 150 professor, maybe they can explain it better. So I apologize for that.
Okay, it's okay.
All right, so let's go on to, if we were comparing a cold-blooded reptile with a warm-blooded mammal at 10 degrees Celsius, what would you expect in these results? How would their metabolic rates compare?
Endotherm will have a higher metabolic rate because endotherm have a higher metabolic.
Yeah, so really what we're comparing here are, yes, an endotherm, the mammal, and an ectotherm, the reptile. And the mammal is always going to have a higher metabolic rate because it has to, it has to generate its own body heat to create that homeostatic environment inside. And at 10 degrees Celsius, that's pretty cold for a reptile. So their metabolic rate would be way slowed down. It would be very, very like nearly hibernational, I think, for most reptiles, because their internal environment temperature is going to match that external environment. Because they, generate body heat. Is equal that of the environment. Okay. Okay, so now it's giving us this data here. It said that rate of respiration was measured in both crickets and mice at 2 temperatures. So 10 degrees Celsius, that's pretty chilly. 25, that's like normal room temperature. And okay, so this is what we found, mils of oxygen per gram per minute for these two different rates. So according to this data, the crickets at 25 degrees Celsius have greater oxygen consumption per gram of tissue than the crickets do at 10 degrees. And we see the opposite trend in the mice. So the difference in these two trends of oxygen consumption is due to what?
See,
Right. It's about that internal temperature regulation. Here we go. Okay, so because the mouse has to generate its own body heat, it's going to have a higher metabolism at a low temperature. And as the temperature warms up, it doesn't have to work as hard to keep that internal body temp going. So that's why it decreases. In the cricket's metabolic rate is always going to kind of match the temperature. So as temperature increases, so does its rate of metabolism. And I basically just explained this answer, but okay, let's put this into words. So the relationship of metabolism with surrounding temperatures. I will say it this way. So as temperatures increase, metabolism increases. So we have this positive correlation between those two variables. Endotherms, it's the opposite, right? So as temperatures increase, metabolism decreases. And it asks us to explain our answer. Well, I can just kind of get it from here. Let's see, internal temperature. Okay. All right, so for the cell respiration part on your exam, you're definitely going to want to memorize that formula for calculating metabolic rate. So mils of oxygen consumed per gram or per hour per gram. And then definitely know the relationship between metabolism or metabolic rate, that is, and temperature, and especially how that's different in ectotherms versus endotherms. And then also knowing the specifics of how that cell respiration experiment were done, and kind of like the purpose of like the water bath and that potassium hydroxide in the chamber. So knowing those little details, that's going to be important on your exam. All right, let's keep rolling to photosynthesis. This is our last topic for this review. And once we're done going through like the review page, I don't mind sticking around for a little bit longer to answer any other like questions you have. about your exam. I'll try to answer them the best I can. All right, so what is photosynthesis? So how do we kind of define this process? And where is it taking place within a plant?
It's the process that plants and other crops use to produce food, crops.
So it's a process that plants use to produce food. We'll say chemical energy. And what do they use for that? What's the photo part there of photosynthesis?
Food.
From the light.
Yeah, from light. So we're making chemical energy from light energy, which is pretty freaking awesome. So yeah, so photosynthesis is creating chemical energy from light energy. And where does it occur? Which part of the plant?
Chlorophyll.
Yeah, so the chloroplast, so that kind of belongs down here where it's talking about organelles, but those chloroplasts are located in the leaves. You could also call it the mesophyll. That's like more like technical term for that tissue in the plant. So yeah, like the green tissues of plants are called mesophyll, and that's where we find those chloroplasts. And as we established before, the cell respiration, which plants also do, takes place in our those mitochondria. So our cells only have mitochondria, but plant cells have both mitochondria and chloroplasts. So they can do both processes. They can do cell respiration or they can do photosynthesis. All right, so let's put out this, our summary chemical equation. We know, we kind of already said that we need light energy, right? What else do we need for photosynthesis?
Water.
Water.
Yeah, we're gonna need some H2O.
Carbon dioxide.
Oxygen.
Absolutely, carbon dioxide. Oxygen is going to be one of our outputs, right? That's actually a waste product for plants, which really benefits us. And then what are we producing here?
Sugar.
Yeah, so we're producing sugars, usually in the form of glucose. Oh, I didn't even see that they had this like word bank here. It's fine. All right, so the leaf extract, when you did that experiment of like separating the pigments, how many pigments did you find?
4.
Yeah, and can you name those 4 pigments?
Chlorophyll A and B.
Chlorophyll A, chlorophyll B.
Carotene.
Carotene, and the one that is a tongue twister, really hard to spell sometimes.
And it's located in the thylakoid discs.
Yeah, absolutely. So that last one, that's the xanthophylls. And yes, the pigments are located in the thylakoid. They're actually embedded in that thylakoid membrane. And that is where they just kind of sit and absorb that light energy. So what colors of light are absorbed most by plants?
Violet and blue.
Yeah, so we got, what is going on? Okay, violet, blue, and then at the other end of the spectrum, the red. I don't know why that text is so small.
180 to 450 nominate.
So you were giving me some wavelengths, it sounded like. Could you repeat those?
I was saying 380 to 450.
Okay, yeah, so 380 to 450, that's our, like the violet blue ones. Violet blue color light. And I'm not, what are they wanting us? Oh, I think right here we just put best. It is absorbed best. And wavelengths of this color contain highest what?
Energy.
Yes, yes. So the shorter the wavelength, the higher the energy that it contains. So
Does plant pigment absorb all colors?
I'm sorry, I missed the first part. I heard observe all colors.
Yeah, does plant pigment absorb all color or not?
No, it does not. So the plant pigments, they really only absorb the like the violet blue end of the spectrum and the red end of the spectrum. And then like the middle portion where you have green and yellow, really, it does not get absorbed at all. It gets reflected, and that's why we see leaves as green most of the time. Oh, hey, and that's the answer here. Okay, so chlorophyll is green because that wavelength is not absorbed. It is reflected. So that light energy just kind of like bounces off of the chlorophyll and back out to the observer, us, and that's why it looks green to our eyes. And okay, so why is photosynthesis important to sustenance of life? What two things does it provide all other organisms?
Oxygen.
Yeah, it provides oxygen and
Sugar.
yeah, sugar, food, organic fuel, however you want to express it. But yes, it sustains us not only by giving us things to eat, but also providing the oxygen that our cells need for cell respiration. Okay, it asks us about the absorption spectra. So that's that one graph that has multiple lines, and it shows like the different pigments and what wavelengths they absorb the best. And that's basically, I mean, what the absorption spectra tells us is what wavelengths are absorbed the best, which we kind of already named up here, that it's the violet blue end of the spectrum and the red end of the spectrum. That's where we see those two peaks in the absorption spectra. That was the 380 to the 450, and the other end, I think it's like around like 620, 630 or something. Oh. Well, let's just say like 600 to 640, I think. I don't have it memorized. I think it sounds right. Maybe it's higher. Now I want to go look at my absorption spectra. Let's see. Okay, yeah, it looks like it's between look, 660 is that red end. Okay, so now we're looking at the that chromatography, the separation of the pigments. So what factors affect the movement of pigments? There were 3 factors involved there. So what are those factors?
The RF distance, equal distance of the pigment, the distance of solvent, which is equal to 17 centimeter per square 20 centimeter. That's
Okay, yeah, so you're giving me, yeah, the RF, how we calculate the RF value. So yeah, that's the distance of the pigment traveled by the pigment divided by, yeah, the distance of the solvent. But what determines how far a pigment can travel? There's 3 factors that affect pigment separation.
Size
Mhm.
of pigment, solubility
in the solvent. One last one, it has to do with the paper.
Yeah, affinity for the paper.
Right, yeah, affinity for the paper. Exactly, yeah, so the size, the molecular weight, or the size of the molecule, how soluble it is in the solvent, and the affinity for the paper, which is kind of like how attracted it is to the paper. Okay, so here it tells us that we found that a solvent traveled 20 centimeters from origin and a pigment molecule traveled 17. So we already have this formula written out, which is another one that you'll have to memorize for the exam. So we're looking at 17 divided by 20. Sure thing. And so
0.85.
I'm sorry, could you repeat your answer? I did not hear that.
0.85.
0.85. Thank you very much. All right, so what we mean by solubility in the solvent, like how readily does it dissolve in that whatever chemical you're using? So for instance, in the lab, you used petroleum ether acetone. All right, petroleum ether acetone, that is a non-polar substance. If you had used water, those pigments would not move at all because these pigments are non-polar molecules. They would not interact with water. They would not dissolve very well in water at all. So the solubility in the solvent, how readily, is it kind of going to interact with those solvent molecules will determine whether it can get like dragged up the paper or not. So because these are non-polar molecules, the pigments are non-polar, you want to use a non-polar solvent. So anything that's kind of like alcohol-based, would be appropriate there. So that's what we mean by solubility in solvent. And then the affinity for the paper is, yeah, it just refers to like how readily does it like stick to the paper. So if a pigment has a high affinity for the paper, if it likes the paper a lot, it's going to kind of like stay put where it is. It's not going to travel. So you want it ideally to have a low affinity for the paper. So then it will be able to kind of move along. It's not going to be like stuck to the paper. It can like travel through it or up it. So like this pigment that we're talking about down here, where we calculated this RF value, 0.85, that's a pretty high RF value. And so if there is a high RF value, we can kind of make some assumptions. So a high RF means that the pigment is a smaller molecule. It is highly soluble in the solvent, and it has a low affinity for the paper. Yes, exactly. So if it traveled a great distance in this nonpolar solvent, that would indicate, yes, that this pigment is nonpolar. So yeah, if you used water instead of this petroleum ether acetone solution, the pigments probably wouldn't move at all. They would have a very low RF value. And if you use a different type of paper, I don't even know like what the paper, I'm not sure. I don't know a lot about paper chemistry, but like if you use a different type of paper, it's possible that the molecule would just like stick right where you put it in that origin spot and it wouldn't travel. So a high affinity for the paper would be bad for this like migration of the pigments. Okay, so let's talk about how we calculated the rate of photosynthesis. So this setup, this was like the Elodia plant in the tube of water, and then you put a big lamp right in front of it. So what did we measure there for rate of photosynthesis?
Measure the volume of liquid expelled out of the pipette and divided by the time it took.
Right, so we measured the movement of that water along the pipette over time. And the way that, or how quickly that water line moved indicated what? What was being produced in that tube by the plant?
Carbon dioxide. Not
carbon dioxide. So we're talking about a plant here that's doing photosynthesis. So it's going to produce one of two things. It's either going to produce oxygen or it's going to produce sugar. Or I mean, it produces both, but which one were we measuring?
Oxygen.
Right. Yeah. So we were looking at those little bubbles that were coming up, and over time, or to see how quickly we could get 0.1 mils of oxygen gas in that pipette tube. Oops. Oxygen. And why did we use this sodium bicarb in the tube? So we had not just water there. Of course, we need water, right, for photosynthesis to occur. But then we mixed in this sodium bicarbonate into the water. So what was that all about?
Carbon dioxide.
Yes, that was our source of carbon. Yeah, so there was no carbon dioxide in that tube because it was completely filled with liquid. So we needed to get a carbon source in there in order for photosynthesis to kind of keep rolling along. So that was our source of carbon, which is just basically baking soda dissolved in water. Okay, and then this was data that was collected. So we had these lamps with different wattages, and then the time that it took to create 0.1 mil of oxygen. So rate of photosynthesis, we're talking about mils of oxygen per hour. So in order to calculate that, we need to convert these minutes into hours. So if this rate equals 0.1 mils divided by, how do we convert, how many hours is 30 minutes?
0.5.
Okay.
Yeah. So 30 minutes is half an hour, so it's 0.5 hours. So then what is our rate calculated as?
0.2 milliliter per hour.
Great, yes, 0.2 mils per hour. Excellent, and then for this one, so 15 minutes is how much of an hour?
Zero 0.25.
Yes, ma'am, 0.25 hours.
0.4.
Great, yes, 0.4. Okay, what about 7 minutes? Yeah, that's what I got too. 0.11 hours.
6.
Well, 0. So 0.1 divided by 0.11. Oops.
0.86.
I got 0.9. Well, I guess it depends on how far out you carry this decimal. I got 0.9.
9. Okay.
So an hour. But again, as long as you're kind of writing out the in-between step, not just your final answer, you'll be fine. So even if your value, if your end value, it like varies a little bit, as long as you wrote your calculation, I think your professor will be fine with it. So this was 0.13.
0.7.
Great, yeah. 0.7 mils per hour. All right, so what is it? I want us to make another graph. Oh, geez. Okay, well, we're not going to make the graph, but it is interesting to note that we see a peak production of oxygen, or sorry, yeah, a peak rate of photosynthesis here with 200 watts, and then it starts to decrease a little bit beyond that. So if we were to graph this, what would be on the x-axis? What would be on the y-axis? So which,
amount of oxygen produced?
right, so amount of oxygen produced would that be X or Y?
Light's gonna be X and rate's gonna be Y.
So the rate, our y-axis is going to be dependent on, yeah, the wattage of time, or sorry, the wattage of the lamp. Yeah, so our graph would be, yeah, like wattage down here, and then along the y-axis, our rate. And so then we would be able to kind of visualize exactly where this peak photosynthesis is happening. And that,
So Professor's graph maybe the line graph.
yes, this would also be a line graph. Mhm. Okay, so this brings us to the end of the review packet. So if there's anything else that you want to go back, revisit, or if you have any additional questions. Yes, and to answer your question, it would be a line graph because, wait, is there? Yeah, so it would be a line graph because in between these values of wattage, there are an infinite possible number of wattages there. So we only looked at 50 and 100, but between 50 and 100, well, there's, you know, a 51 wattage, there's 52 wattage, there's 52.35 wattage, there's, you know, so there's like an infinite number of possible wattages between these values that we have chosen. So yeah, once again, our independent variable is both quantitative and continuous. And yeah, if you're finished up and you don't have any additional questions, you're welcome to sign out or like log off. Have a great evening and a great rest of your
Thank you
week.
so much, Professor. Good night.
You're welcome.
Bye, thank you. Good night.
Good night.
Thank you so much, Professor.
You're welcome.
Professor, good night.
All right, so page 5, yes, I can go back to page 5. Oh, that right, this was our